Hello everyone, myself Pavalan, 2nd MSc Applied Microbiology, Sagadat College, Tirupattur. We are going to see about bioremediation of pesticides. Definition: a pesticide can be defined as any substance or mixture of substance invented for preventing, destroying, repelling, or mitigating and pest. Pesticides like insecticide, herbicides, fungicides, and various other substances are used to control, inhibit plant disease and insect pests. The positive aspect of application of pesticide renders an enhanced crop food productivity and drastic reduction of vector-borne disease. However, excessive use of these chemicals leads to the microbial imbalance, environmental pollution, and health hazard. Criteria for biodegradation. For successful biodegradation of pesticide in soil, following aspects must be taken into consideration. First step, organism must have necessary catabolic activity required for degradation of contaminant at fast rate to bring down the concentration of contaminant. The target of contaminant must be bioavailability. Soil condition must be congenial for microbial plant growth and enzymatic activity. Cost of bioremediation must be less than other technologies of removal of contaminants. Strategies for biodegradation. For the successful biodegradation or bioremediation of given a contaminant, following strategies are needed. Passive intrinsic bioremediation. It is the natural bioremediation of contaminant by title indigenous microorganism and the rate of degradation is very low. Biostimulation, practice of addition of nitrogen and phosphorus to stimulate indigenous microorganism in soil. Bioventing, process of biostimulation by which gases stimulates like oxygen and methane are added or forced into the soil stimulate microbial activity. Bioaugmentation, it is the inoculation introduction of microorganisms of the contaminant soil facilitate biodegradation. Composting, piles of contaminated soil are constructed and treated with aerobic thermophilic microorganisms to grade contaminants. Periodic physical mixing and moistening piles are done to promote microbial activity. Phytoremediation can be achieved directly by planting plants which hyperaccumulate heavy metals for indirectly by plant stimulating microorganisms in the rhizosphere. Bioremediation, process of detoxification of toxic unwanted chemicals, contaminants in the soil and other environment by using microorganisms. Mineralization, complete conversion of the organic contaminant to its inorganic constituent by a species or group of microorganisms. Different approaches for biodegradation. Bacterial degradation. Most bacterial species degrade pesticide. Most of the pesticides undergo partial degradation leading to the formation and accumulation of metabolites. Fungal degradation. Fungi degrade pesticide by introducing minor structural changes to the pesticide, rendering it non-toxic and are released to soil, where it is susceptible to further biodegradation by bacteria. Enzymatic degradation. Enzymatic have a great potentially to effectively transform and detoxifying polluting substance because they have been recognized to be able to transform pollutants at a detectable rate and be potentially suitable to restore polluted environments. Chemical reaction leading to biodegradation. The biodegradation of pesticide is often complex and involve series of biochemical reaction. Detoxification, conversion of the pesticide molecule to a non-toxic compound. A single change in the side chain of a complex molecule may render the chemical non-toxic. Degradation, the breakdown of transformation of a complex substrate into simpler products leading finally mineralization. Example, Thyrum fungicide is degraded by strain of Pseudomonas and the degradation products are the dimethylene proteins, subpolites, etc. Coagulation, in which an organism makes the substrate more complex or combines of pesticide with cell metabolites. Conjunction of accomplished by those organisms catalyzing the reaction of addition of an amino acid. Organic acid methyl ground to the substrate. For example, the microbial metabolism of sodium dimethyl diethyl carbonate. Activation. It is the conversion of non-toxic substrate into a toxic molecule. For example, herbicide 4-butyric acid and the insecticide porate are transformed and activated microbiologically in soil to give metabolites that are toxic to weeds and insects, changing the spectrum of toxicity. Some fungicide pesticides are designed to control one particular group of organisms, pests, but they are metabolized to yield products inhibitory to entirely dissimilar groups of organisms. For example, the fungicide PCNB fungicide is converting in soil to chlorinated benzoic acid that kill pests. Leaching. Since many of the pesticides can be solubilized, they are removed by leaching. Advantage. Often less expensive and site disruption is minimal. It eliminates long-term liability. It can be coupled with other physical or chemical treatment methods. Disadvantage. Treatment time is typically longer. Rate of contaminants that can be effectively treated is limited to compounds that are biodegradable. The process is sensitive to the level of toxicity and environmental condition is the ground. If the process is not controlled, it is possible the organic contaminants may not be broken down fully, resulting in toxic byproducts that could be more mobile than initial contaminants. Thank you for listening this video.